So the virus we're going to be talking about is a little complex as viruses go. And it's complex also in terms of the nomenclature, which some wise scientists decided to give the name COVID-19 and then SARS-CoV-2. It's one and the same thing. The disease is COVID-19. The virus is SARS-CoV variant 2. Now, SARS was a respiratory disease that was first identified in 1993, and since then there hasn't been a major outbreak again. But nearly 9,000 people died from SARS, and this virus COVID-2 is very similar to SARS, as you'll see when we show you the genome. It's very similar, and in fact, it's in some ways scarily similar. And people have asked, could this occur again? And I think the answer to that is yes, because there are 5,000 approximately viruses of this type in nature. And so with that huge number of viruses in nature, I see no reason why you can't get recombination occurring again. The recombination is an event that occurs at high frequency with these viruses. And we have to watch out, I think, that in fact we've got more bad news wrapped in protein. This was a comment made by a very famous scientist, Peter Medawar, who was an immunologist who won the Nobel Prize. And he said, in fact, in an article he wrote, that it's simply a bad piece of news wrapped in protein coat. And that's really what it is. SARS itself, if you look on the left-hand side, is an enveloped RNA virus. So the genomic information is contained within a piece of RNA, and it's positive strand RNA. And the RNA is protected from outside forces by the nuclear protein, or the N protein, which hides the genome inside it. Then there's the matrix protein or membrane protein, depending on which language you speak, whether you want it to be matrix or membrane. And this protein, again, protects all of the genome from external forces. There's a small integral membrane protein that's known as E. Its function really isn't very clear, but the major glycoprotein that this virus has in its surface is the spike glycoprotein S, which is a trimer. Now, this is an interesting protein because it actually has some analogies to the Ebola virus glycoprotein and also the influenza virus hemagglutinin. It structurally has some similarities which are worth noting, that these proteins, in fact, are similar to each other. And if we look at the sequence of these proteins, which is shown here, we can see that, in fact, that the S protein is a trimer, which has a globular head and a stalk. And then the hemagglutinin neuraminidase is called HE. Not all coronaviruses have an HE protein. Only a few of them do. And so we have to be careful that the third glycoprotein is only specific for a very few number of viruses. There's a matrix protein, as I said, thank you, Curt, that spans the membrane. And then there's the envelope glycoprotein E that also spans the membrane. The nucleocapsid protein is shown here, round up. It actually has its crystal structure solved in several cases. But the crystal structure of the other proteins is known for some of them and not for others. And there's no complete picture where, for instance, TGEV, which is and mouse hepatitis virus, have these proteins, and they're found quite abundantly. Can we have the next slide, please, Curt? Let's go back. Let's look at the phylogeny of these viruses. We've got some of the viruses listed here, and that is the hepatitis 229E is originated in bats, probably, and then had an intermediate host. Now, one thing that's quite common among these viruses is the need for an intermediate host. That's very true. We don't know that if that's true yet for working with our viruses, but we do know it's true for SARS, which in fact has bats as an intermediate host, and then MERS. MERS is a virus that comes through camels, but is a coronavirus nonetheless. And all of these viruses are related to each other, and you can see that in black put around the diagram is images of bats, humans, civet cats, and etc., and camels, which are the hosts, intermediate hosts for these viruses. Next slide, please, Curt. Now, one of the things that's absolutely essential for doing virology is the ability to be able to do reverse genetics. That is, to make mutations within the genome and ask what effect does it have on the replication of the virus. And for coronaviruses, there are 3 or 4 very specific approaches that can be done for reverse genetics. On the top left, you can see SARS Urbani. Urbani was a scientist, an Italian scientist who worked for WHO and actually died of SARS, one of the first people to die of SARS. And so the virus was named after him, whereas now we have this nomenclature system where we try and avoid people's names or places, and it becomes very complicated. Hence, it's the SARS-CoV virus that we're dealing with now. The methods for doing these genetic approaches are state-of-the-art genetics, and that's you can clone fragments of the genome in back vectors. You can ligate together fragments of the genome with T7 RNA polymerase, and you can clone fragments into vaccinia, another virus, big genome DNA virus, which will take, in fact, quite a lot of extra nucleotides. And so you get fragments cloned into vaccinia, and that's been quite successful in terms of being able to do reverse genetics. The ability to break the genome up into pieces like this, and then, in fact, to be able to manipulate them in the laboratory has been a very useful proposition for, in fact, making various forms of mutant virus, and also, in fact, for hopefully at some point, eventually making a vaccine. Next slide, please. Our phone's ringing here. So the virus uses as a receptor a variety of proteins. Probably the most important is the ACE2 protein, which is the protein that, in fact, binds, well, first of all, AC1CT. The ACE2 protein binds to this cellular protein and acts as its receptor. There are other viruses that use CCAM1 as and it binds to sugars on these viruses. And there are 1 or 2 other minor components which also act as receptors for the virus. But the important thing is the receptors are sufficient for getting entry of the virus into cells. Next slide. So I'm going to summarize the talk that I've given in the last few minutes to say that the virus binds to a receptor on the cell surface. It then enters the cells via endocytosis. And this may have stimulated probably what I suspect is bogus use of drugs that raise the pH, intracellular pH. The genomic RNAs the genome RNA gets translated in the cell. You get fragments of RNA made. They're very large, some of them, 1A and 1B are huge pieces of RNA, and that gets translated into proteins. And these are non-structural proteins. Then the helicase transcriptase complex gets formed and it forms a complex that gets transcribed. And I think I'm due to stop there, aren't I, Curt? Oh, one more. The virus has a double membrane in virus infected cells, and this double membrane is visualized by cryo-electron microscopy, and you can see there, leave it there, and there you can see is that the double membrane that's formed, and that's quite unusual because it's only SARS-like viruses that in fact can get these double membranes from cells. And it's a complex mechanism by which they get that from cells. Now I'll let Curt continue.
Okay. All of the viral RNAs, the genomic RNAs of the coronaviruses, have very highly conserved leader sequences and transcriptional regulatory sequences. You can see they form these RNA secondary structures that are highly conserved between the different viruses and even in the SARS-CoV-2. And I think these are maybe some interesting areas where an intervention can take place using RNA-directed editing or targeting techniques. Right, so Bob described this entry process for SARS-CoV-2. It binds the ACE2 receptor, enters the cell, and then uncoats the RNA. And I'm going to tell you just briefly about the replication of this virus. So there's a lot of detail. Some of the resources that Andrew has put into the folder I think can be particularly valuable. All right, here's just a comparison of what came out in 2003, known as SARS virus, or people are calling now the classic SARS, versus the new virus that's out now, the SARS-CoV-2, as it's called. You can see from the genome structures illustrated here, these are very similar. In fact, the new virus, if you look just at the N protein, this nucleocapsid protein here, or the nucleotide sequence, has 88% identity to current BAT isolates that are out in circulation, almost 80% identity with SARS virus, SARS classic, and 50% identity with the Middle Eastern respiratory syndrome. And when you look at the amino acid sequence, of course, it's even higher conservation. The virus genome itself is a positive strand of RNA, and it's about 30 kb in length. And so when that positive strand of RNA gets into the cytoplasm, it engages a ribosome, and it gets starts to be translated starting at the 5' end. The first thing that gets translated, as Bob mentioned, are these things called ORF1A and ORF1B. These are two open reading frames at the 5' end that both of them encode polyproteins. The ORF1A differs from ORF1B by a -1 frame shift, but the virus actually has engineered a way to make the ribosome translate the polyprotein AB by, in a percentage of the cases, causing a stem loop and slippery sequence form a pseudoknot that causes a force to make the ribosome backtrack in order to gain access to the -1 frame shift and produce this entire protein. It's about 800 kilodaltons worth of polypeptide being synthesized all at once. But actually the business parts of these proteins are the small non-structural proteins embedded in the polyprotein. And they're liberated co-translationally by first this papain-like protease, PL-PRO, which is encoded by the NSP3. And second, this cysteine-like protease 3CL that's encoded by NSP5. And they cleave up this polyprotein into about 16 or so individual polypeptides that can together form the RNA-dependent RNA polymerase that's the replicase and transcriptase complex for this virus. like many RNA viruses, the coronavirus, as Bob mentioned, replicates inside some protective vesicles in the cell. Sometimes for some viruses, these are single invaginations into organelles. For the coronaviruses and flaviviruses, for example, they use double membrane vesicles that are derived from the endoplasmic reticulum Golgi apparatus intermediate complex called ERGIC or ERGIC. It's not really clear how these double membrane vesicles are formed, but what happens is that the genomic positive strand RNA, as I mentioned, gets translated to form the replicase complex. And the actual site of replication is within these vesicles. The transcriptase forms a complete copy of the 30,000 nucleotide genome, copying it to negative sense, but also, and then that serves as a template for copying positive sense, and those replicated genomes are exported back out to the cytoplasm where they can assemble. In order to assemble, they need to make these structural proteins as well. What I've talked about are only these ORF1ab products, which are non-structural proteins, but Bob mentioned the spike and other proteins that are encoded by these open reading frames. The virus has a really unique mechanism to access these. It forms 3' collinear messenger RNAs, and those all have the identical sequence as the leader, forming as a cap-like structure on the end. And there are different ways that the virus can access this leader through discontinuous transcription, either from transcribing a copy of the antigenome, starting at the leader and accessing the downstream gene by a looping mechanism, or by copying the genome itself and making an antigenome mRNA that forms the subgenomic mRNA. All of these mechanisms have been shown for different viruses, so whether it's one that's preferred for CoV-2 or not needs to be determined. Finally, the virus assembles in the cytoplasm and buds from the cell surface. There's not a lot of information on virus budding for many viruses or any viruses, but as Bob mentioned, the nucleocapsid protein coats the RNA, serving as like a histone to protect the RNA. The packaging takes place where the structural proteins in the membrane and the matrix protein assemble with the nucleocapsid in order to form a budding virus. This is really an important area for intervention. For example, the influenza virus drug Tamiflu, neuraminidase inhibitor, prevents virus from budding off and being released from the site of replication, and that has a big role in containing the virus infections. If you just bear with me for a few more minutes, I just wanted to talk about something closer to where my research is focused, and that is the response of the host to the virus infection. The coronaviruses infect the cell and deliver their RNA genome and the replication intermediates into the cytoplasm. And the cell has cytoplasmic proteins that are designed to survey and sense foreign nucleic acids, such as structured or a 5' triphosphate, or perhaps the structured leader sequences of this virus genome, are recognized by these RNA sensors and turned into a transcription activation signal that produces cytokines like interferon, the primary antiviral mediator, as well as many other antiviral genes that get regulated by IRF3 and NF-kappa B transcription factors. The interferon also and other cytokines such as interleukin-6, which is very important in the COVID-19 clinical presentation. They bind to receptors and mediate JAK-STAT signaling pathways that also produce transcription factors such as the STAT1-2 complex called ISGF3. And it's this complex that makes hundreds of mRNAs that turn into proteins that are antiviral effectors that protect cells from virus replication. The problem is that clinically, interferon and other cytokines can be both good and bad. From classic SARS, there were a couple of presentations that were of interest. Patients were found that gave an early interferon response. So just like the textbook, the interferon is induced at the first sign of virus infection. The interferon reduced the virus titer. There was a modest levels of inflammation and very mild clinical disease. On the other hand, there were patients that had no interferon signaling, so the virus titers were very high. But in these cases, they had a little bit of a greater disease, but it was mild compared to patients that had delayed interferon responses, where the virus titer was high, the interferon came on too late to control the virus, and greater disease was seen because of dysregulated immune responses at the inappropriate time. So it will be very interesting to analyze the data from the current outbreak, especially the clinical presentation, and see what differences there are in cytokine responses that drive the acute versus mild diseases of COVID-19. Just a few comments on the druggable targets. These proteases, as I mentioned, the RNA polymerases, these proteins, unlike the spike protein, do not vary and are highly conserved between SARS, MERS, and SARS-CoV-2. A lot of the nucleotide analogs that you hear about, such as remdesivir, are causing chain termination from the RNA-dependent RNA polymerase. And then many of the other drugs that have been discussed are kind of summarized in this slide. It's available in your Google Drive folder if you guys want to see it. Again, mentioning some of the things that Bob mentioned, like the chloroquine that's been popularized by some politicians, also IL-6 inhibitors that have been effective in the clinic. There's already been a bit of work on this, and some of you may have seen the bioarchives, have been reading it, pulling out papers. This is just one example of a paper from Krogan's group that is in the preprint server. They've made protein-protein interaction networks for all of the non-structural proteins of SARS-CoV-2 and some of the structural open reading frames. They've used that to identify druggable targets that are interacting with these proteins, and did some network modeling of drug target interaction space as well to try to repurpose existing drugs. There's a lot to research there. And this is probably too much for me to read to you, but Dan had asked us to identify some areas we think are important. As before I had really seen what you guys had already suggested. Obviously, we're at the, compared to the 2003 SARS epidemic, there's going to be a huge amount of data generated and also tools that have been created in the decade and a half since then to analyze these data points. Remember, SARS was at the beginning of deep sequencing, and now we have so many technologies. But I also find it very interesting, some therapeutic challenges like distinguishing pre-symptomatic people who have very high virus titers who are spreading disease from uninfected, how to diagnose future severity for individual patients, or even weaponizing antibodies that are available that are not neutralizing by themselves. The symptoms of the acute respiratory distress are important. And obviously preparing for the next pandemic, we had SARS, MERS, flu, Ebola, Zika. Why weren't we prepared for COVID-19? Obviously, this is an area that we need a lot of thought. And consider the possibility that the next epidemic or pandemic might not be a human pathogen, but an animal or plant pathogen that would really cause severe consequences for people. And the example I like to give is the famous Irish potato famine, which killed over 1 million people. So COVID-19 has nothing on this plant pathogen. And it destroyed the economy in a way that has really never been recovered. So thank you very much for listening to me and to Bob.